Good morning, grade 8. We are now in the continuation of our topic. Okay. Let's move now to cell cycle. By the way, for your project this school year, are you familiar with your project this school year? Okay. Don't worry, I'll just give you ample time to do your project. Your project for this school year is all about learning resource. Okay, learning resource or resources material. So what's in the learning resources material? Okay, create a learning resource material, okay, out of our topic this fourth quarter. Our topic for this fourth quarter is cell division and genetics. So out of cell division and genetics, all you have to do is to create a resource material or learning resource material. What is a learning resource material? This is anything that will actually explain the concept or science concepts, okay, in our fourth quarter module, okay, module 1, weeks 1 to 4, right? This includes but not limited to digital outputs, for example, blogging and video documentaries. This is narrated. And physical outputs like the 3D of science concepts, okay, diorama, miniatures of science concepts, the actual science concept if possible. Okay, I am encouraging everyone to please wait. Going back, again, I am encouraging everyone to please make a physical output. Okay, why? Since it has the biggest grade, okay, at the same time, it will be exhibited when we are allowed to do so. So make it as attractive as you can, okay, or as possible in your own creativity, materials of your choice, in any materials of your choice. So that is the overview of your project. Again, in our concept, in our science concept now, in our topic, there are a lot of possibilities, there are a lot of projects that we can actually generate, okay? Because the DNA is there. What else? Cell cycle is there, so you can do anything about it. You can actually create something out of it, okay? So that's it, okay? It could be, again, digital output, which is blogging, which explains the concepts, of course, and video documentaries narrated. That's for the digital. But if it is physical, the 3D of science concept, three-dimensional, okay, diorama, miniatures of science concept, and the actual science concept if possible. Okay, let's move now to our topic. Let's move now to cell cycle. Okay, eukaryotic cells undergo an orderly set of events which culminates in the production of new cells. The target for the cell cycle is the production of new cells. Okay, that's our main goal. Okay, or the cell cycle. This set of events is called the cell cycle. It is divided into 4 distinct phases. We have G1, okay, S, G2, and of course the M phase or the mitotic phase. G1, S, G2, and the M phase. Okay. The control of cell cycle is influenced by both external and internal factors. What are these? Nutrients and growth factors, as well as the cell size, may trigger a cell to divide. The cell cycle is a complex process involving special proteins that control the switching on and off of these 4 phases. Okay? These control points assure that the cycle is progressing as it should be. Okay? CDK, okay, take note, or the cyclin-dependent kinase, it is an enzyme, one of the major control switches that signals the cell to proceed from G1 to S, or from S to G2, or from G2 to M phase. Okay? That is actually one of the signal enzyme. This is an enzyme. Okay. Another substance called P53 is a protein that blocks the cycle if the DNA is damaged. Take note, it blocks the cycle. It blocks then the cycle if the DNA is damaged. Why? Because it actually prevents what? Manufacturing defective cells. If DNA is too damaged, this may lead to cell death or apoptosis. Okay. Recent studies reveal that the P53 is frequently mutated in human cancers. This is the reason why cancer cells continue to what? Progress. Metastasis. Okay? That's the spreading of cancer cells. Cancer results when the regulation of cell cycle goes out of control and normal cell growth and behavior is lost. Okay? This is the layout of the cell cycle. Okay? As you can see, G1, it started with G1. This is the growth and normal metabolic roles. Okay? In this phase, it functions, the cell functions what? In their normal metabolic roles, metabolic processes. Okay? That's the first phase. The second phase is S phase. S phase is the synthesis phase. This is where your DNA replicates. DNA replication happens here in preparation for M phase or the mitotic phase. The third phase is the secondary growth phase. This is the growth and preparation for cell division. The cell needs to prepare itself for possible cell division. That is why after DNA replication, okay, G2 now prepares the cell for possible cell division. So what are the things that G2 is actually doing for preparation? Okay, it prepares the cell for the nutrients needed for cell division. Number one. Number two, it prepares the cell for the splitting of cells or cytokinesis. Okay, the materials needed for this splitting of cells. Okay, here comes the next phase, which is the mitotic phase or the M phase. In the mitotic phase, you can see what? The 4 phases of mitosis. The prophase, metaphase, anaphase, and telophase. Sure, according to our books, some of the books includes interphase. The interphase is the preparation. Okay, interphase includes G1, S, and G2. That's interphase. That's how the cell actually prepares itself. Okay, that's interphase. Prophase, metaphase, anaphase, and telophase, these are the phases of cell division. Cytokinesis is the what? The splitting of cell or two daughter cells. Okay, let's continue. This may result to tumor and plum cells that grow and divide abnormally. Some tumors, though, are harmless or benign, such as fibroid cysts in the uterus, but it can become malignant too. Okay? Benign and malignant, what are these terms? When you say benign, it is mild. Okay? It cannot actually cause any harm, but it can be malignant. Malignant is harmful. Okay? And it can harm an organism, which means that the tumor grows uncontrollably and destroys surrounding and nearby tissues. And as more tissues are destroyed, it can cause death of an individual or organism. Cancer cells differ from normal cells in that, number one, they divide more rapidly. Remember that the cell division is not that faster as you think. Okay? Cell division needs ample time to happen. The cell needs to prepare. Here comes our what? Cell cycle. Okay? Cells need to prepare for possible cell division. But in cancer cells, they divide more rapidly. Second, they tend to wander and more about and invade other tissues. They wander and more about again, invade other tissues. They tend to change the DNA characteristic of the nearby tissues. The third one produce different surface proteins as compared to the parent cell it came from. Meaning to say, if it produces more surface proteins, therefore, the genetic characteristic of these cancer cells is not exactly the same of the parent cell because it carries what? Another cell. Or it actually what? Modified itself. Exposure to mutagens, mutagenic substances, or agents that causes genetic changes like X-rays, gamma rays, carcinogen, cancer-causing substances, activation of oncogenes. We have all oncogenes, by the way. We have all cancer cells, but it is not activated or inactivated. Okay. How can we activate or how these oncogenes be activated? By exposing ourselves to radioactive substances, by exposing ourselves to mutagens, or by exposing ourselves to carcinogen like your soft drinks. Your soft drinks is a carcinogen. Okay. What else? Your preserved goods, it is actually one of the carcinogen. It is an activator for oncogenes or genes with a potential cause or cause cancer-causing potentials, or a combination of these have been linked to cause cancer. Okay. The first three phases prepare the cell for cell division is by duplicating its genetic material and producing organelles and cytoplasm for the future daughter cells. That is actually in preparation. So what I'm telling you. The initial phase, which is G1 or GAP1, is the first growth phase. Okay, during this time, the baby cells grow and prepares itself for DNA replication. Okay, enzymes and proteins necessary for DNA replications are also produced during the later part or the latter part of G1. The S phase or the synthesis phase is a time when the cell replicates its DNA. Okay, why do we need to replicate the DNA? In order to copy the genetic characteristic of the parent cell. Okay, the identical chromosomes appear thin and thread-like as the chromatin fibers that make up the chromosomes are still uncoiled. It is not actually untangled. It is still tangled. It is not coiled. It's uncoiled, and you can actually see that it actually thickens itself. As soon as the cell divides, cell will not proceed to the next phase. If DNA has been completely replicated, centriole also divide at this point. So the centrioles now prepares also itself for possible cell division. The next phase is the G2 or the GAP2. This is the second growth phase in which the cell manufactures necessary materials, the one that I'm telling you. It will need when the cell divides, such as the organelles, materials for new membranes, and cytosol. When these materials are ready, the cell then proceeds to M phase or the mitotic phase of cell division. Okay, together G1, S, and G2 make up the interphase stage of mitosis. This is the interphase with the preparation stage. Ironically, while some describe interphase as the resting stage, the cell during this time is in fact very busy preparing the cell for cell division. Of course, because again, if the cell is not ready for cell division, therefore, it may create a different set of cells. Okay? It may create tumors, it may create cysts, which is a defective cell. The M phase or mitotic phase is a process in which the nucleus divides into two nuclei. Okay? Each new nucleus has the same number of chromosomes as the parent cell. A parent cell contains two complete sets. Okay, two complete set of chromosomes. We consider this one as diploid. It is not haploid because it is complete. So meaning it is diploid. It commences with prophase, which consists of stages for both nucleus and cytoplasm. The nuclear envelope breaks down, the nucleolus disappears. The duplicated centrioles begin to move from the opposite poles and form spindle fibers between them, and the chromatin fibers become visible as they condense, coil, and become shorter and thicker. Okay, I will let you see what prophase, metaphase, anaphase, and telophase would look like under the microscope. The duplicated chromosomes held together by a centromere now appears more distinct, and the two identical chromosomes or sister chromatids begin their journey to the equator or at the center. Okay, they're attached to the spindle fibers by way of the special structures in the centromere called kinetochores. That is prophase. Okay, let's move now to metaphase. During metaphase, chromosomes migrate randomly and line up towards the equator. Again, if the chromosomes, if you're going to look at the microscope, under the microscope, if the chromosomes are in line in the at the center or the equatorial line, meaning to say that that cell is now in the metaphase. Okay, another checkpoint occurs at this stage when the chromosome is not properly aligned at the mitotic spindle. Mitosis will be arrested, or meaning stop. Okay, this is to ensure that complete set of chromosomes will be distributed in each new nucleus. As a result of this checkpoint, sister chromatids do not separate until the complete set has been organized and ready to be passed on each daughter cell. Anaphase. Let's move now to anaphase. What is the anaphase? In anaphase, the sister chromatids that make up the duplicated chromosomes separate and move towards the opposite ends. Now, if you're going to look at it under the microscope, you can actually observe that the cells now is moving away from each other. Okay, once its sister chromatids separate, it is now distinct, now a distinct chromosome or new distinct chromosome. The migration is made possible because proteins that make up the spindle fibers contract and pull the chromosomes apart towards the opposite end. Okay, this is anaphase. In telophase, this is the last stage of or phase of cell division. Nuclei form in both poles as new nuclear membrane forms around the nucleus from the remnants of the parent cell. And the membrane materials built during the G2 phase. Remember the G2 phase? Again, in the G2, we are actually preparing the cell for possible cell division and the materials to use in the cell division. Now, we are now using the membrane materials that are actually built in the G2 phase. Because, again, we are now separating the cell. We have two distinct daughter cells with exact number of chromosomes. Nucleus reappears and spindle fibers begins to disappear. Okay, this last stage of mitosis is somehow the opposite of prophase. Okay, exactly opposite of prophase. Right? Cytokinesis, this is the division of the cytoplasm. It happens, begins in the mitosis end in the late telophase, and is completed when the cell contents have been divided equally to the two daughter cells produced, that is in the mitosis. Okay. In animal cells, the cytoplasm cleavage or cleaves to form a new cell. Okay, it actually forms a cleavage. In plants, however, a plant or a cell plate forms at late anaphase between new developing nuclei. Centrioles are also absent in plant cells because plants don't have centrioles. But similar structure that does the same function is present, which produces spindle fibers. Still, it has spindle fibers, but it doesn't contain any centrioles. Mitosis may not be accompanied by cytokinesis, resulting in cells to have more than one nucleus or two nuclei. It will become multi-nucleated. Okay, sometimes cytokinesis doesn't happen. Okay, that is the reason why if you're going to look at under the microscope, you can actually see two nucleus there. Okay, some of the plants or plant cell or animal cell has two nucleus. Okay, so this is now the layout of cell cycle and cell division. As you can see, you're going to observe, this is in G2. you're preparing the cell. Okay, this is the preparation stage. Okay, after DNA replication, okay, G2 now prepares the materials needed for prophase or M phase. This is the M phase or mitotic phase. In prophase, as you can see, okay, the nuclear membrane starts to dissolve itself. Spindle fibers appear. Okay. In metaphase, okay, it is actually aligned at the center. As you can see, the chromosomes now are at the center of the equator, and the spindle fibers is actually attached to the centromeres and the kinetochores. Okay, this is the spindle fibers. Okay, right. The next phase is the anaphase. It actually separated. They're actually pulling apart from opposite end. As you can see, they pull each other apart. Okay. This is your centrioles. Your centrioles pull the chromosomes or sister chromatids apart. Okay. And this by the help of the spindle fibers. And the telophase. Telophase is the final stage of cell division by which it forms a cleavage, okay, going to cytokinesis for the forming of new cells, two new daughter cells. As you can see, the envelope now is starting to form. Nucleolus actually reappears, okay. Spindle fibers disappears, okay. And of course, cleavage is formed in animal cell. In plant cell, it has cell plate. Okay. And afterwards, it will become two daughter cells. Okay, let's stop with mitosis. Hopefully, you learned something out of our discussion. Do you have any questions so far? Question with mitosis or cell cycle? All right. I think that will go for today, class. Again, for your project, I will just give you an information. I will update you in your LMS. I think that will go for today, class. Have a nice day. Take care. Bye-bye. Have your lunch. Enjoy the rest of the day. Bye-bye.